So, and with that, Jane?
Yeah, great. So I'm really excited to be able to introduce Jamie, and it's always fun when you sort of have a relationship with the speaker. And I remember, I think the first time I actually met Jamie or sat down to talk with him was at a conference in autism, and I was a little nervous because I thought, oh my goodness, I've read his work. He's so well established. Is he going to ask me questions about EEG, of which I know very little? And all of my fears were allayed when we sat down and realized we had a shared affinity for IPAs. So that's always a good way to start off, is when you sort of have things in common, not only, research, but, personal. So, and since then we've continued to share IPAs on occasion, but also to sort of work together. And he's actually a consultant on one of our studies, really helping us to navigate the challenge of collecting EEG on children, preschool age children who have developmental disabilities, which is a near impossible task. But, thanks to his help and his models for a lot of what he's done already, we've been successful. So I've really enjoyed working with him. And he's one of those people that, is not only friendly and easy to talk to, but like he'll take I've learned he'll take a breath. And when he does that, it's like, okay, he's going to shoot me some really sort of tough question. I better brace myself to answer it. So he has an intellectual rigor to keep me on my toes, and yet also delivers in such a, very supportive, wonderful way. So anyway, without further ado, officially, Jamie is an associate professor of child psychiatry and psychology at the Yale Child Study Center. He is also director of the Developmental Disabilities Clinic and of undergrad studies. He teaches a seminar to the undergrads on neurodevelopmental disabilities, which I think is incredible. I'm sure that's their favorite class. His research really focuses on advancing biomarkers to support intervention research, and he has been continuously funded for 15 years plus. So, and he also has many awards that testify to the impact he's had. So without further ado, I want to turn it over to Jamie, and excited to hear what you have to share.
Thank you, Jane. What a very, very nice introduction. And I feel similarly. And it's always nice to have colleagues that you can do great science with and have a lot of fun with. And, for those in the audience who, you may hear this talk and you may think Jamie will never be the scientist that Jane is, but Jamie will forever have the CPDD bar shuffleboard title that he took from Jane. So I have that, if nothing more. Okay, let's start. All right, so I'm going to talk about progress in biomarker development in autism. I'll talk a lot about a consortium that I'm involved with, the Autism Biomarkers Consortium for Clinical Trials. These are the groups that have funded my research and that pay me, and there's no conflicts, I don't think, with anything that I'll talk about today. This is what I want to talk about. I want to talk, hang on, I want to move my control bar so I can see my own slides. Great. I want to talk about where things stand in biomarker research in autism, what I think some of the important goals for biomarker research in autism are, what I see as important scientific and also practical considerations. And then I'll walk you through some examples of some of these kinds of attributes with my favorite biomarker, an EEG marker called the N170, and then talk about some challenges I think that we have to overcome before we really get to a place where these are viable. And then maybe some ideas for how to get them viable, talking about strategies for better research in the context of the Autism Biomarkers Consortium for Clinical Trials as an example, and then also ways to maybe use these biomarkers to think about ways to create new treatments. So let me start by giving you a quick summary of the kinds of tools that we use in the lab. And one important takeaway that Jane mentioned in the introduction is that I'm a neuroscientist, but I'm also a clinician. And so I think sometimes about the mismatch between those settings. And really, I think, we're actually further along in the science than we are in the clinical science in some regards. And some of the work that I really, the thrust of the lab to try to bring some of the clinical standards to some places. So as an example, in the lab, we do lots and lots of EEG, and you'll hear me talk about lots about that today. we mess around. We're not good at it, but we mess around with positron emission tomography by collaborating with people who are. Same thing with NIRS, functional near infrared spectroscopy, and then we do lots of eye tracking as well. And so what a bunch of cool and powerful tools that we have as scientists to try to understand autism. But then when I change gears and I walk across the street to the clinic, what tools do I have at my disposal as a clinician? I have literally the exact same tool that Leo Kanner used in 1943, right? We have our eyes and our clinical insight. And we can do nice things like structure it around a standardized play session or things like that, but we don't have anything that's objective. We don't have anything that is more sensitive than what I can see with my own eyes or a parent can see with his or her own eyes. And that, to get that, we want biomarkers. Biomarkers are, they can mean lots of things. The FDA and NIH convened a group really to think about what a biomarker means. And this is the definition that they came up with: a defined characteristic that's measured as an indicator of normal biological processes, pathogenic processes, or responses to an exposure or intervention, including therapeutic interventions. So really, I mean, there's a lot of words there, but if you think about it, what is a biomarker? A biomarker is something that you can measure objectively that gives you meaningful information about a biological process, right? And this is something that we don't have in any reliable and usable way in autism presently. Why do we want them in autism? So these are just some broad categories. So in that, if you're interested in biomarkers, I would encourage you to you see it referenced there, the best resource. It's lots of information, and it puts autism biomarkers in the context of broader biomarker research, because there's a lot of fields that are much further along than we are in thinking about what biomarkers are. And in fact, I mean, it's not part of this talk, but if we want to get into during the question and answer session, people in autism, I think there is a field-wide fundamental lack of understanding about what biomarkers are and how to evaluate them. But so these are some examples that are in this best glossary. Like a prognostic biomarker would be something that tells us about the future course of a disease, right? So this might be something that we can measure that in a young child with autism, that tells us what their IQ will be like later, that tells us what their language might be like later in life. We could think about a pharmacodynamic or a response biomarker, and this is something that tells us that a particular system, for us, we're thinking about neurodevelopmental conditions, so maybe a brain system, it would show us that brain system has been engaged by a treatment. And then we could think about, and this is, I think, what most people in autism reflexively think about with biomarkers, are diagnostic biomarkers. And this can mean a few different things. So I think that the conventional way to think about a diagnostic biomarker is of the condition, right? That a person with this biomarker or a biomarker in a certain range has autism. And that's, that to me is a really lofty and maybe unattainable goal, because autism isn't a thing, right? Autism is many different biological entities that just tend to look similarly. So when we talk about measuring biology and finding something that is not just common to all those etiologies, but definitional of any one of these etiologies, it's possible, but it seems ambitious to me. A different way to think about a diagnostic biomarker is to think about it as diagnostic of a subgroup. So another way to put this would be to think about like a stratification biomarker. This figure is from a paper written by Eva Loth and colleagues a few years ago. And really the idea is basically you have this really heterogeneous group of people with autism. Maybe a biomarker could help them sort them into subgroups in ways that are meaningful for long-term outcome, for treatment selection, things like that. And we really don't have things like that. And the work that I'm talking about today is really geared towards that kind of goal, to try to be able to stratify the really diverse group of people with autism in ways that are practically useful. In terms of biomarkers for autism, there are different sets of concerns for lots of different kinds of biomarkers. As I describe some of the things that I think are important to you, I want to emphasize that any given biomarker doesn't need to be able to do all of these things. So when I just gave you 3 examples of different kinds of biomarkers, right? Like the attributes that one would require for a good prognostic biomarker are not going to be the same attributes that one would require for a diagnostic biomarker. And so you can see a given biomarker, it may have a profile of strengths and weaknesses in some of these attributes that could render it well suited for one but poor for another, but it's not an all or nothing. A biomarker that fails to do some of these things can still be useful in certain contexts, and the FDA term is context of use. When you evaluate a biomarker with the FDA, you define a particular application for it, and you're only asked to provide evidence for its utility in that particular context. So some of the things that I think are worth understanding about biomarkers are whether they're sensitive to diagnostic status. And I don't mean to contradict the idea that we're not looking for diagnostic biomarkers, but even if a biomarker isn't definitional of autism, if it doesn't show some difference between autism and typical development, low odds that you're barking up the right tree. We would expect, again, similar idea, doesn't have to associate necessarily with the diagnosis. It could associate with symptoms. Autism measures lots. Autism touches upon lots of different things. So maybe there are biomarkers that are going to associate with language, and biomarkers that are going to associate with cognition or social function. And these all may map onto diagnostic categories in different ways, but they also may relate to those domains of function in typical development. But we should see relationships between the biomarkers and those aspects of symptomatology. We also conversely would want to see that they aren't necessarily generically specific. And let me give you an example. If I have a biomarker that I think is indexing social function, but it's actually telling me something about cognitive function, it's going to be tough to notice that difference because cognitive function is going to put a rate limiter, in a sense, on social function, right? But if I did an intervention that actually improved social function, it might, but my biomarker is measuring intellectual function, it's not going to move, right? And so we really need to be clear about what the biomarkers are measuring, but also what they're not measuring. And we have to be alert to the possibility that we could think our biomarkers are measuring something just because it's confounded with a third variable. We want biomarkers, or what we don't necessarily want, we have to consider whether biomarkers in autism are applicable across development, because autism can apply to 60-year-olds and it can apply to 6-year-olds. And do we, if we have biomarkers that are useful in autism, can they be the same across the lifespan? Do we need to conceptualize biomarker batteries for different developmental cohorts? So that's something to think about as we evaluate biomarkers. We want biomarkers, depending on the biomarker, that are, we want to understand how they're affected by behavior. Isn't always true. If you have a genetic biomarker, doesn't matter what the kid is doing when the blood is drawn. For those of us who do EEG biomarkers, right? If we're interested in a biomarker that's telling me something about the social perceptual system, and the child is having a tantrum while I'm trying to record the EEG, well, I'm getting meaningful information, but it's not meaningful information about the social perceptual system, right? And so we need to think about what's happening during the biomarker acquisition and whether that could be skewing the biomarker values. We would like biomarkers, maybe, depending on the context of use, to be sensitive to change in clinical status, that as a kid gets better or worse, we would see the biomarkers values correspondingly change. And so these are the kinds of things that I think about as being important to understand from a scientific perspective in evaluating biomarkers. But then I also think there are practical considerations, right? Because I don't want to develop biomarkers that are useful in my lab. I want to develop biomarkers that are maybe useful in large scale clinical trials, or biomarkers ideally someday that we could use in clinical practice. And to think about implementation in that way, it also puts practical considerations into play. Like, can we do this in a reasonable way in people on the autism spectrum? Can we do it in an economically feasible way? And how accessible is a technology? Right. So if we had a super duper crackerjack biomarker, but it could only be done in autism centers of excellence, this is how many people we could affect, right? But if we had a biomarker that could be rolled out in any hospital, this is how many people we could affect. And this is what we want. We want to be thinking about biomarkers that have this kind of reach. And so We, myself, and I know there are lots of EEG friendly scientists in the crowd, use an awesome technology in some of these respects. For those of you who know a lot about EEG, you can ignore it the next minute. For those of you who don't, EEG is the electroencephalogram. It's brain activity recorded from the scalp. Generally speaking, we record two kinds of EEG. Resting EEG, which is kind of the brain's oscillatory idle, and task-related EEG, in which we let discrete things happen, sights or sounds, and by doing it repeatedly, it lets us tease out what of that electrical signal is actually yoked, is tied to the cognitive process that we're inducing. There are great things about EEG for this kind of work. EEG requires wearing a wet hat, so it's pretty tolerable as far as brain imaging goes. So we can use it in very young children. We can use it in people across the intellectual span. It's non-invasive. It's pretty movement tolerant. If a child is moving during an EEG, you're going to, probably lose the trials during which they're moving around, but it's not going to ruin an entire recording session. So all things considered, it's a really good technology for working in this population. When we think about some of the other things I've mentioned, EEG is also really practical. EEG, it's expensive to buy an EEG system, but for there's lots of different ones. For the one that I've depicted here, the cost of acquiring data is salt water and latex gloves, right? So once you have a system, it's really cost effective, and they're also accessible. This EEG system isn't everywhere, but there's an EEG system pretty much in every hospital in the country, and we already use EEGs at the population level for screening, for seizures, for newborn hearing. And so it's a technology that, should we find things that are beneficial, could be deployed at scale in a feasible way. Also, for autism, and you can study autism in lots of different ways. As will be evident in my talk, I tend to focus on the social communicative aspects of autism. As I've said several times, autism is a tremendously heterogeneous disorder. As a diagnostician, when I give someone a diagnosis of autism, you can only know one thing about them, that they have difficulties in social behavior and communication, right? It's truly the only universality in the diagnostic criteria of autism. And so that's where I tend to do my research. And that's also a place where EEG has been very effective, even in typical development. So we have a nice foundation of understanding, to look into a poorly understood neurodevelopmental condition. This is my favorite ERP. The ERP is event-related potential. So event-related potential just means that you process the EEG signal in a certain way. The way I described before, you make repeated things happen. So you get this, what's called a waveform, that tells you something about a particular cognitive process. So can you all see my, my cursor? Okay, great. So this purple swath is highlighting this negative deflection in this waveform. So time is going this way in milliseconds, voltage is going along the y-axis, negative is down, and this is the N170. It's called that because it's negative going, and it happens at around 170 milliseconds. This is what it looks like in typically developing adolescents and adults. And what this marks, it's a really robustly studied ERP in typical development, indexing what we call structural encoding of the face, the stage at which you recognize a face as a face prior to, this is a happy face, prior to, this is mom's face, this is a face. And this was actually the first study that I did when I was a graduate student, and we wanted to see really whether we would see an N170 in people with autism, given their difficulties with gaze and the poor face recognition that had been seen in behavioral studies. And what we found is that they did have an N170, but it was different from their intellectually and age-matched peers in terms of being slower, which we interpreted as less efficient. And so this is a finding that has borne out across many studies that I'm going to tell you a lot about over the next several slides. But as I go through, I'm going to put the studies that I discuss in the context of those kinds of scientific challenges for biomarkers that I described. So this first study did give us a piece of information about this N170 as a potential biomarker, right? That its latency is slower in autism. And so it does this is not at a per person level. You can't tell who has autism and who doesn't based on their N170 latency, but we do see it hangs together with diagnosis. We looked at in this first study, behavioral performance in terms of face recognition, and we found that people with autism made many more errors in face recognition, and we found that a person's performance associated with their N170. And so again, what we found is when we looked at the social stuff, we saw the same pattern of results that we'd seen before. We saw that people with autism made more errors on face recognition tasks. We saw that their N170 is slower. Contrasting this with the information that we saw to the okay, so again, referring back to my list, so we've seen now, kind of cool, not necessary, but kind of cool, that the N170 is something that's there from when a child's about 3, and we're seeing the same pattern of differences really in children, adolescents, and adults. So developmental applicability of the biomarker. And then we looked at non-social stuff. We saw behaviorally no differences. Kids with autism read and decoded phonemes as well as their typically developing counterparts. And in terms of their brain response, we saw normal specialization. We saw, these purple bars are the letters, the green lines, the pseudo letters, and we can see everyone is showing a strong, this is actually amplitude, not latency. We see that everyone is showing the specialization marked by increased amplitude, and we didn't see any differences in latency between the groups. And so we're seeing this N170 that is slower with people with an autism is slower specifically to faces. It's not that just like the sensory system is working more slowly. It seems to be specific to faces. Next thing we wondered was what's happening when we do an ERP experiment. So we know lots of eye tracking studies have shown us that people with autism tend to look less at the eyes of the face than typically developing children. But we also know that the N170 latency is modulated by facial features, and you get the fastest N170 to the eyes. And so what if our idea that this is telling us something different about the brain in autism is really just saying, no, their brains maybe work the same, but when you sit them down and do the experiment, they don't look at the stimuli the same way, right? But that's still meaningful, but my gosh, we don't need to waste energy netting them. Then we could just do an eye tracking experiment, right? But we wanted to look at this. And so what we did was we used a manipulation where we had a crosshair that preceded the faces. So it's not the way it's depicted here, in that the crosshair didn't obscure the face. It was there, it disappeared, and the face appeared. But what we did was prime attention to look at the eyes, the nose, or the mouth. And we wanted to see is if we force people with autism to look at the eyes, so that the same kinds of visual attentional patterns as the typical developing children, would the latency delays go away? And they don't. Really, the opposite. So these are data you see typically developing children shown in the darker colors on the left of each pair of bars. Attention to the eyes is shown in blue, nose shown in red, mouth shown in green. And really the takeaway is that overall, people with autism tend to be slower. We see that this is most pronounced when they look to the eyes. So rather than looking to the eyes, normalizing the N170 in people with autism, it's making the difference between them and typically developing peers more salient, right? It does not fix the problem. The N170 delays are not an artifact of where they're looking on the face. Make them look to the eyes. It would actually be a better experiment. You'd get more pronounced N170 latency delays. The last thing that I'll talk about kind of the scientific properties of the N170 is, and this is really preliminary in 2 regards. 1, not yet published, and 2, very small sample, I think 7 kids. But we wondered how the N170 would change if children's symptomatology or clinical status changed in the course of a treatment. We partnered with Pam Ventola, who is doing pivotal response treatment at the Child Study Center. This is a behavioral treatment for autism. It's under the umbrella of applied behavioral analysis therapies. It's well supported by research. It's geared to improve social function by virtue of naturalistic and kind of play-based teaching. And so what Pam did was she was doing research in preschool age children. They would get a 14-week course of treatment. they would get better, their symptoms would be reduced. And we did N170 assessments before and after. And what we found is, so this is data for each individual child. So you can see 7 lines and a dotted line that's showing us the average. The pre-treatment assessment is on the left, the post-treatment assessment is on the right. And you can see that all of these kids, except on average, and for all of the kids except one, their N170 gets faster as they get better in treatment. So, again, preliminary, but suggestive evidence that the N170 latency changes alongside clinical status. And so if we review what we've talked about so far and put it alongside the stuff that I mentioned at the start, we see that the N170 is sensitive to diagnostic status. It associates with symptoms. It associates with symptoms in a functionally specific way. It doesn't always just correlate with a generic set of processes. It's applicable in children, adolescents, and adults. It's robust to behavior in terms of at least gaze behavior during our experiments, and it seems to be sensitive to change in clinical status. If we also think about some of the other things I mentioned earlier, it's nice that it's viable in this population, it's cheap, and the technology that we use to derive it is accessible, right? So awesome. But we've got a long way to go before the N170 or really any other biomarker is ready for use as a kind of an outcome measure or a stratifier for clinical trials, or certainly in clinical practice. And why? Well, first I want to make clear that this is a story so far about the N170, and actually so far it will remain a story about the N170. And that's not because the N170 is the only thing that's relevant. It's because what I know best. But there are, there's lots and lots of evidence for other biomarkers, eye tracking biomarkers, other EEG biomarkers, MEG biomarkers, fMRI biomarkers. But the criticisms that I'll make of the N170 apply to all of these biomarkers. One is that there's limited reproducibility. It's not always true. So at the bottom of the slide is, I think, maybe, I don't know if I've updated it this year, but all the studies that have looked at the N170 since we published that first study in 2004. And a cool one is this Kang meta-analysis from 2017, which actually looked across all these studies and said, hey, you know what, that latency delay that they saw is true across these studies when you do a meta-analysis. But it doesn't mean that every one of these studies has found the same thing. There are studies in this mix that have had no results. And so what does that mean? Does that mean the N170 is that our findings are false? No. It could mean lots of things. It could mean that in a condition as heterogeneous as autism, maybe we shouldn't expect the same biomarker pattern for everybody. Maybe some of these samples have preserved face recognition or preserved face processing, and so the N170 wouldn't look different. That's a good problem to have because we could study it. We could tease it apart. There are other problems that are harder, that are true of this literature, that are true of most bodies of literature in autism. Many of these studies are small, are underpowered, so may have found no result just because they weren't adequately powered to find them. That's a bigger problem, right? We can't really undo that problem in the literature or fix it with future studies and bigger samples. And lastly, we don't know how variability in methodology affects things like this. There are tons of choices we could make when we do an EEG study in autism like this. We could use color faces, black and white faces. We could have a crosshair or no crosshair. We could use my EEG system or a different EEG system. And we really have no idea whether any of those decisions could influence your pattern of results. And so there's tons of variability in that regard in this literature as well. There are other problems endemic to biomarker research in autism. For most biomarkers, we don't know how reliable they are within a person, and we don't know whether the act of taking repeated measurements alters biomarker values. And these are really things that are critical to know given the drive to use them in clinical trials where there will be definitionally more than one assessment. Lastly, we don't have, for most biomarkers, any kind of normative reference. Right? By contrast, if I was interested in head circumference, I could download the CDC growth charts and know exactly where a person with autism stands on that biomarker. There's no growth chart for N170, right? And so our inference in any given sample is really specific to the control group in that sample, which doesn't put us in a position to make large-scale inferences. So what I think we need, and what I've been trying to do for the past few years, is research that addresses these kinds of issues. And really, I didn't know I was doing it at the time, but what's become evident to me over the course of the past 5 years is to get the kind of data that we would need to qualify these biomarkers with the FDA so that they could be used as a tool in a broad way in clinical trials research. So what would a study designed to address these issues look like? Well, it would take well-evidenced biomarkers. So this sounds obvious, but if you write grants, those of you who write grants will recognize that it's really, really hard to get the 41st study of the N170 funded, right? You need to be doing something that's innovative as the first. But what we really want to do is interrogate the biomarkers that have the strongest evidence. We want well-characterized cohorts so that we can understand the relationship with individual differences. We want big samples to power us to make inferences about individual differences, and including in typically developing kids, so that we get that kind of atlas, that reference that we want. We want longitudinal designs, and not longitudinal in the sense that we think of human development, but longitudinal in the sense that we think of clinical trials, like that we have a sense of test-retest reliability, and we have the sense of stability over the course of like 4 to 6 months of the duration of a typical clinical trial. We want methodological rigor so we can understand and eliminate noise as much as possible. And then ideally, we want to use practical assays so that if we figured something out, we could deploy it at scale. And this is really the set of considerations that was in play when an RFA was put out now 5 years ago to initiate the Autism Biomarkers Consortium for Clinical Trials. And so the ABCCT was a multi-site naturalistic study. I won't go through all the details on some of these slides, but I'm happy to share the PDF if people would like afterwards. But it was a big study of 280 children with autism, 119 typically developing children, school-aged, variable IQ from moderate to mild intellectual disability to well above average, using EEG and eye tracking with a longitudinal design and with a blood draw. There were things there about the study that were pretty unusual or unique. One, it was a grant mechanism that brought together a governance that involved academics, but then also government scientists at NIMH, NINDS, NIDCD, and NICHD, and then also industry people. We adopted a level of rigor that really, truly is unprecedented in 3 areas. One, in terms of regulatory, we administered it as if it was a clinical trial, though it wasn't. Methodologically, I mean, really everything down to the stimulus presentation monitors across sites were identical. And then statistically, so that we really pre-registered every hypothesis down to the level of a single dependent variable with a directional hypothesis. So really, to be clear, going, when the results come out of this, saying, it's not like we dug around and found something interesting that we thought we knew we were going to find, and either it worked or it didn't, which is a benefit if you're going to bring things to the FDA. And this is the battery of biomarkers that we use. We made one EEG biomarker primary and one eye tracking biomarker primary. The ERP, you could have guessed it, is the N170. The eye tracking is a composite across 3 different tasks that really all involve looking to human beings or faces in scenes. We also did the gauntlet of clinical measures, the status quo that we currently now use really to understand outcome in clinical trials and autism research. The idea maybe that we can do better, and also to let us see how our biomarkers relate to the phenotype. And the study worked. We were successful. We met our milestones and got people through on time. The hypotheses for both of our primary variables worked out. The N170 latency, as you can see here, was, as we would have predicted, slower in people with autism. We saw that we get pretty good signal. We got valid signal in 97% of the typically developing people, 76% of the people with autism, which may or may not be a problem. Happy to discuss during question and answer. And we saw what we classify as adequate stability. So if we correlate a person's response baseline with their response 6 weeks later, about 0.66 in ASD and 0.75 in TD, and we see relationships between the N170 and the phenotype, strongest with face memory, and we'd seen before. The eye tracking composite, which we call the ocular motor index, similarly, we see less looking to faces in autism. We see higher reliability, about 0.8. We see near perfect acquisition for eye tracking. It's such a user-friendly technology in this population. And we also saw relationships with the phenotype. So this was good. This was good. And so with this outcome, we submitted letters of intent to the FDA's biomarker qualification for both of these biomarkers. They were both accepted, one in May 2019, one in March 2020. To have a biomarker letter of intent accepted to the biomarker qualification program doesn't mean a heck of a lot. It means, well, I'll take that back. What it means is that basically means that they're interested, that these are good data, that they think this shows promise. What it doesn't mean is that it's a qualified biomarker. It means that they're asking you to take the next steps, to prepare a plan for how you would acquire the additional data that would be needed to qualify it as a biomarker, and then eventually to undertake the final step would be to submit a biomarker qualification package, which they would then thumbs up or thumbs down in terms of whether the biomarker is viable. Now, the reason I say, so obviously we're not there yet, but the reason that I hesitate to say not a heck of a lot is because this is still an important milestone. So these are the first biomarkers accepted into this program for autism. They're the first biomarkers accepted into this program for any psychiatric disease. And this is really, this is a cutting edge for us, and it's a cutting edge for the FDA. Really, the field has not yet finished figuring out how to conceptualize biomarkers for psychiatric conditions. And so it's a really interesting and fun space to be in, where we're kind of working with the FDA to make sense of this kind of information. As I mentioned earlier, when any given letter of intent is specific to your proposed context of use, we propose this as a diagnostic biomarker, not diagnostic of autism, but diagnostic of a subgroup of people with autism that you might use to enrich clinical trials by reducing heterogeneity. The idea that this is the OMI distribution, so these stacked histograms, you can see if I just crudely draw this purple swath, there is a group of people with autism who really don't overlap at all with the distribution of typically developing kids. And so like a biomarker like this is not going to be useful to say autism versus not, because you can see the vast majority of people with autism are within the same range as typically developing people. But maybe this group has some kind of common neuropathology or more heterogeneous kind of phenotype, or maybe is more likely to have a common response to a treatment. And so that would be a stratum that we could include in clinical trials for enrichment. And really, that's where things stand. We have 2 grants from the FDA, basically to support collaboration with them in discussions to figure out exactly what kind of data would be most useful to figure out how these biomarkers could be used in clinical trials. It's not straightforward. It's interesting and it's complicated how we would determine a cut point. How do we, when we believe that our clinical measures are inherently subjective, inherently unreliable, and problematic in other regards, how do we establish the ground truth for how that purple group may be different? And then lastly, how do we make sure that these findings don't only count if you're using, an electrical geodesics it used to be electrical geodesics, a MagStim geodesic sensor net, right? How generalizable they are to other things. So those are stuff that's very much in process. We're very excited that we were renewed in July. So we're going to go on for another 5 years. The studies, there are 3 studies that are involved in the renewal. A confirmation study where we will basically do our statistician doesn't like to use the word replication, but that's really the way, that would be the term that we would most use to think of it, that most of us would use to think about it. The same study again, 200 skewing the proportions, so there's more TDs, 200 ASD, 200 TD, same design, same battery, really just eliminating the biological motion paradigm that didn't do much in the first study. The second study would be a follow-up study where we would bring back the original ABCCT cohort. This would end up being 2.5 to 4 years post-enrollment, maybe actually a little bit longer given delays related to COVID, and to let us look at the long-term stability of these markers, how they change when children, one problem with the first study is the 6-month span we had in 6 to 11-year-olds. We didn't see a ton of improvement or worsening of symptoms in that span of time, but over this longer period of time, maybe we would, to let us look at biomarkers and how they track those changes. And then also to let us look at longitudinal predictive value. So how do biomarker values acquired, 4 years ago, do they tell us something about how a person will be functioning presently? And then the last study is a feasibility study, really to see whether we could do this battery in younger kids, 3 to 5-year-old children. And I'll mention one more thing, and then we can talk about questions and answers, is just to show that there's more in my life than the ABCCT. One of the things that we're working on is trying to figure out whether we can use some of these ideas to develop new treatments. And really, the way that we're thinking about it is, proof of concept before therapeutics. But the idea, we have an idea of the brain regions involved in generating the N170. We have an idea that they're also involved in governing some of the patterns of visual attention we see in our eye tracking studies. We see from other research, from fMRI research, that these brain regions like the STS, the superior temporal sulcus, change activity levels when a person benefits in behavioral treatment. So maybe, having seen those things passively, maybe that is telling us something that this could be a target for stimulation. And so some of the work that we're doing now really is targeting with transcranial magnetic stimulation, the STS, to see whether we move the needle with the N170 or whether we alter patterns of visual attention to faces. And just as again, very preliminary teaser, in a pilot sample of typically developing controls, we're hoping to do a bigger study. A grant is actually under review now. We see that even in these TDs who don't have any kind of social difficulties, that this stimulation to the STS makes their N170 faster, and it makes them look more to the eyes of the face. And so, suggesting that maybe this could be something that's useful. And I'm going to stop there. I do want to provide some acknowledgments. The families that work with us, as I mentioned, I'm a clinician, and the work that we do in the lab is interwoven quite tightly with the work that we do in the clinic. And so these are the clinicians involved in the Yale Developmental Disabilities Clinic. The ABCCT is a very large group. This slide just highlights leadership, and then the group of people who do the work in the lab, and I'll stop there. Thank you so much. And I can see there are some questions already. Were the kids followed as they age to see if N170 remains stable after that initial treatment? Has anyone tried the same behavioral treatment in adults and adults with slow and see those change? So I'm assuming this is talking about the preliminary data with the N170. So we did not have a follow. Maybe we have a follow-up like on 3 kids, not enough to know anything. We don't know that if it would stay stable. I don't know. I mean, it's a good question. I don't know. Yeah, I don't know. And there's not data that I'm aware of from our lab or other labs to speak to it. There's a study by Jerry Dawson using ESDM and looking at ERP indices of face perception that showed after treatment more normal patterns of, it wasn't the N170, of other ERP components. It's a tricky study in that that study had no pre-treatment data. It just showed that like in kids who didn't have treatment, there were differences. and in a different group of kids who did have treatment, there weren't differences. And so their interpretation is that that the treatment normalized it, but it's a strong interpretation given the data. So that's something we need to understand more about. But it's a good question. And the other question, is there a relationship between N170 and comorbid psychiatric disorders or any other psychiatric disorder other than ASD? Yes, totally. N170 is not, N170 disruptions are not specific to ASD. You see differences in the N170 in anxiety, in social anxiety, in schizophrenia. We've done one study that we're analyzing the data from now, comparing adults with autism to adults with schizophrenia. And what we found is that the N170, the latency delays were evident in the autism group, but not the schizophrenia group. But I think of the N170 not, again, not as like a diagnostic biomarker of autism, but as an index of functioning of a neural system that's really relevant to autism and central to autism, but could also be relevant to other conditions, like, that affect social function, like schizophrenia, like anxiety, for example. What about adults with ASD and anxiety? Are they different? Don't know. We have, we've never done a study where, we've actually tried, we haven't gotten it funded yet, to do that schizophrenia study in anxiety, but we don't. So what I would like to do is really do a study where we do the gold standard diagnostic procedures for autism and anxiety on people with anxiety and people with autism, so that we really have a rigorous characterization of symptomatology. We don't have that. What we do have is like self-report measures, like the BAI, like the Beck Anxiety Inventory and the State Trait Anxiety Inventory. And what we find is really that in our data, and I don't think this is the final story, I think it's yet to be written, and there's literature in the anxiety world that would challenge this interpretation. But what we've seen in our data is that the anxiety metrics actually associate more with a component before the N170 called the P100, which we think of kind of vigilance to the stimuli, or attentional factors, but less on the N170. But these are all really great questions. I mean, these are the kinds of things that I think need to be understood that aren't understood yet. We also don't know in the kids. I mean, like, in the ABCCT, we actually, we really tried to get a psychiatrically clean sample, but so many kids with ADHD or comorbid, I mean, with ASD or comorbid for ADHD, don't have an idea how that might skew things. One of the things we intend to do in the follow-up study is when we see these kids, they would then be between 13 and 16 or 17, and when a lot of the most common comorbidities would arise, like anxiety and depression. And so we intend to collect better data on comorbidity, more specific data on those kinds of comorbidities in the ABCCT follow-up. In the original ABCCT, really our only data on comorbidities was a screen of the child and adolescent symptom inventory.
Great. Are there any other questions anybody has?
Pretty stupid about EEG. I spend my time thinking about molecules. So that question is for the N170. How do you narrow that down to a region of the brain?
Well, it depends on what you think about source localization. So actually, okay, you have who I would put as the best source localizer on Earth at USC, John Richards. So source localization in ERP, to figure out where the source of an ERP is quasi impossible. We can constrain it and we can do sophisticated math. But the other things you can do is build on understanding from other domains, right? Like we know the parts of the brain that are involved in face-related activity, dating back to the 40s from, histology, right? And people who have like prosopagnosia. Then MRI and PET studies that show us that there's parts of the brain, the fusiform gyrus, and the superior temporal sulcus are involved in perception of faces and biological motion. And so what people have done that we have not done, but is administer paradigms, ERP paradigms and fMRI paradigms in the same people, okay, that show that the N170 responses correlate, not perfectly, but they correlate with things like bold signal in the fusiform gyrus. So giving us a sense that this is where it's coming from, but we don't know with great specificity where these signals come from. But I also don't, not to be glib, but I don't care. It doesn't matter where the signal is coming from for it to be telling us something that is meaningful and important for these purposes, right? Like when we do an ADOS, when we do that behavioral diagnostic tool, we have no idea what brain regions are reflected in the things that I'm coding, and yet it's good enough for our autism diagnosis. I think even if I don't know what brain region the N170 is coming from, if it lets us do some of these things in ways that are objective, I would argue much likely be much more sensitive than what I could see with my own eyes, then that's great, even if we don't know where it's coming from. And I think it's a good trade-off. Like the accessibility and non-invasiveness of EEG versus fMRI, I'll take it.
Amy, I have a question. Can you hear me?
Yep.
First of all, thanks for the talk. Every time I see you speak, I get really excited about the state of the science in our field. So my question is a bit broader. I've been really grappling with this sort of argument and debate in the field about the concept of neurodiversity, and that individual differences in things like brain function or social cognition or social abilities are just part of a range of human ability or human function, not necessarily an indicator of disability. And I grapple with this because a lot of this biomarker research is aimed at kind of like fixing the problem, right? Like improving outcomes or improving screening or diagnosis. And I keep going back and forth in this debate, and I really would love to pick your brain and kind of hear about how you reconcile that and how you talk about it with families that you work with in the clinic and individuals that you work with in the clinic, because it's something that really has been just driving me nuts lately.
That's a great question. So I think I have two thoughts. One, like neurodiversity is a big topic. We could talk a lot about it. And if that's the actual question, I'm happy to. But let me answer the more specific question that you asked about how this relates to biomarkers. And I'm going to ask you a question, Abby. I am 5'8". Am I short?
No.
You don't need to answer it. You don't need to answer it. The point is that like short or tall, right, is like a valence is a valence term that we can apply to a biomarker value. But there's nothing that's intrinsically tall or short about 5'8". It's a measurement. And so there's nothing that's intrinsically good or bad about an N170 latency. It's a deviation, right? It's a deviation from another group of people. And so I don't think that these things need to, I think it's a discussion that if we think about it is a very, very different discussion than a discussion about neurodiversity. I think that the idea of whether one wants to fix something or whether one wants to be fixed or whether something needs to be fixed is a totally different conversation than whether one can measure some of these things in meaningful ways, right? I mean, the measurement, and maybe I'm giving myself a free pass, but like we're trying to measure, like there's no harm in figuring out how to measure this, but then it's on somebody else to figure out whether to fix it or not. But I'm happy to answer that too as a clinician. And I think like, it's tough. We as a society, I mean, I think that we can think about neurodiversity in two ways. We can think about kids and we can think about adults. And every adult starts out as a kid, right? But we as a society have a firm policy that children don't get a vote. Like if children, like the idea that we should or should not treat children with autism, like that's decision for the parents. Like, and again, I don't mean to say that like I don't mean to be making light of it, but just think about math. If children, forget about social skills therapy, if children chose whether they study math, most of us would have stopped studying math and algebra, right? So we have decided as a society that parents, adults, make these decisions for the children, right? And so I mean, as a person who works mostly with children, I give the information, I let parents do with it what they will. But the other thing with neurodiversity, it's, I think in general, it's helpful to think of these things as differences rather than deficits. The complexity is that no one person can speak to what is desirous for another person, right? You know, a person with autism can't speak for all people with autism. And the other thing that's tricky too is it's really hard to know among people with autism how they're how to dissociate their history of intervention from their current ability to evaluate the utility of intervention, right? You know, Noah Britton is a person I was speaking on a panel with, and he was saying, you know, they used to try to make me look in the eyes, and it was like torture for me. I wish they hadn't have done it. And an honest question, Noah, like, do you know, are you sure that we would be having this conversation if they hadn't made you jump through all those painful hoops? And he didn't know. And well, no, he didn't know. He said, absolutely, it didn't help. And but I can't know. And so, you know, it's hard to, it's an interesting and difficult situation.
Great. I think we have one more question and then we'll wrap it up. So there's one more question in the chat, Jamie.
Sure, Connor.
About big picture down the road.
So Connor, no. I mean, so to be clear, I'm going to say it again. I don't see the N170 as a diagnostic biomarker of the categorical diagnosis of autism. So I don't think that the N170 is going to be like a post-MCHAT screener. I mean, it doesn't mean that it couldn't be used that way, but none of the data that I've presented today would really be supportive of that, right? Think about those distributions that I showed you, how overlapping they are. I think of it more like saying, like, look, you have a drug that you think is going to help core social symptoms in autism, and you want to do a clinical trial, and you don't know, you know, you only have a set amount of money, and so you can't bring in thousands of participants. So how do you figure out who to bring in? Well, right now, you might say, I'm going to take the people with the highest ADOS scores. Now, you could have a high ADOS score for lots of reasons, right? It could be eye contact, it could be social insight, it could be language, it could be repetitive and restrictive behaviors. I think of things like the N170 as a more granular and more objective way to stratify people with autism for potential inclusion in clinical trials, right? That someday you'd say, like, I want to do a clinical trial, I want to have the most inferential power. How do I get myself a saturated sample of autismness? I'm going to take the people with the longest N170s. That's the way that I think about it. We're not there yet. We don't know, you know, we don't know what the N170 means about autism. I will say that working with the FDA is humbling in that, you know, it's the opposite, you know, the statement I just made, right? I keep saying we don't know, we don't know, we don't know. And that's not the way you write titles for papers published in Nature Neuroscience, right? But that's what it's like when you're working with the FDA. There's no point in blustering or making things that aren't there, because you're all gonna sit there and tease apart the data, and either it works or it doesn't. And so I will say this process has helped me to be very realistic and critical of our data.
Okay. I lied, Jamie. There's one more quick question, if you can answer it quick, or else we can just wrap up. So it's about, can you see it from Stefania?
So Stefania, do you think the N170 I think that yes, the N170 studies in typical development suggest that the N170 is related to configural processing. So I think that for sure, the N170 is telling us something about configural processing. I don't know that the crosshair paradigm would cause people not to adopt a configural processing approach, in that the crosshair is not on the faces it co-occurs. The faces are on screen for like 500 milliseconds. And if a person fixates multiple times during that 500 milliseconds, the trial is probably gonna be paused by eye artifact anyway, right? So they're still taking in, I mean, these faces are not so big that you're only seeing the eyes, nose, or mouth. You're still taking in the entire, I mean, a foveation still lets you take in the face in its entirety. So I don't know that the process of configural processing relies upon fixation to a particular place, like the center, but I don't have any reason to believe it would disrupt configural processing. I mean, the thing we could do is, would be to present figures like facial features in isolation. But again, I mean, we've done lots of stuff kind of thinking about processing strategies, and I love the typically developing literature, Martha Farah and Jim Tanaka. these like all the things that I cut my teeth on working my dissertation. But again, I think about things really practically now. And as long as the same thing is hitting the retina, what I want to know right now is what my ERPs tell me and how that could help me make practical decisions about people with autism. I can't, whether it's because of a difference, whether it's because of a different differential reliance configural versus holistic, like versus piecemeal processing. I don't know that it matters for these purposes.
Great. I think we could spend an hour talking about all these great questions. But we have to end, and I want to certainly thank you, Jamie, for an awesome talk and for giving us a lot to think about. And thanks so much, and especially appreciate you reminding everybody that you are the shuffleboard king at bar. So we will have to work on dethroning you, and the competition continues. But anyway, thanks to everybody for being part of this. You are welcome to join if you're part of USC and want some updates on the USCAND efforts. Otherwise, we'll let people transition off for this part of the talk. And thank you.
Thanks, everybody. I enjoyed that and great questions. Bye, everybody. And Jane, don't take that away from me. That's honestly like my greatest athletic achievement in my life. Leave me that.
Okay.
Bye-bye.
All right. We'll just have to think of another one for me.
Well, it looked like it went quick.
Yeah.
Yeah, in terms of people getting off. So why don't we go ahead and get started so we can get going. So this was our first meeting of the semester. We're going to have another one in December. And at that one, we'll have a few people talking about their work. But just to give you some updates, there are a couple of new faculty I wanted to introduce. One is Layla Larson, who's muted and no camera, but she's in public health. Hi. You want to say a little bit about what you do, Layla, what your interests are?
Sure, yes. So yes, I'm a new faculty in the Arnold School of Public Health. Sorry, my baby's like crying very loudly in the background, so sorry about that. So I'm in the Department of Health Promotion, Education, and Behavior. So most of my work has been in global health. I'm interested in the consequences of malnutrition on early child development and brain functioning. And we've actually just finished a trial in Bangladesh where we've collected EEG on over 300 children. And we'll be looking at the relationship between that and iron deficiency. And I've also been working in Malawi on something similar.
Very good, thanks, and welcome.
Thank you.
And the other is Christy Welshans, who's in biology.
Hi, I'm Christy Welshans. Like Jeff said, I'm in the Department of Biology. My research focuses on Down syndrome, which is the most common genetic cause of intellectual disability. And what we're really interested in understanding is how changes in neural wiring during development contribute to this intellectual disability. We study this in a mouse model of Down syndrome. And then we also have induced pluripotent stem cells that have been created from individuals who have Down syndrome that we differentiate to neurons. And this provides us with a human model to study the cellular and molecular mechanisms underlying this as well.
Very good. So Jane and I spent part of our summer meeting with various administrators, and we were refunded for this year, so which was great. I think it will continue. So we'll cross your fingers. But we made a lot of progress last year, I think. So we made 2 faculty hires. Christy is one. Fiona Hollis, who everyone's met before, joined us last year as another. Hired in our first year at the School of Medicine. Our psychology search, we didn't fill that, so that's going to be going on again this year. Turned out that COVID created a lot of complications for that issue. So the other 2 searches that we are moving forward with, one is a joint appointment, hopefully, between engineering and psychology for someone who's working in the Artificial Intelligence Institute. The other one is that we're working towards is a co-appointment between the school psychology program and psychology and the School of Education. Hopefully that's going to work out. The Dean of Arts and Sciences is supportive. The Dean of Education is supportive. And the Provost seems to be supportive. So we'll give you an update on that at the next meeting. Whether that search will go on this year or next year, we'll see. We awarded 2 pilot grants, and on December 2nd at our meeting, we'll hear about those. One was to Fiona Hollis, David Mott, and Jesse Kluzik. And the other was to Jesse Bradshaw, Amit Sheth, and I'm blanking on the third person that's in Amit's group that was there, Jesse.
Yeah, his name is Uwer, and he is a postdoctoral fellow in the AI Institute.
Good. Great. So as I mentioned, they'll give like a 10-15 minute talk on the objectives of their pilot project. Those were awarded in mid-May or mid-June, I think. But with COVID, things being shut down, we won't be pushing on progress there. We hope we'll have 2 more pilot projects coming up this year. We'll do announcements for those about the same time we did last year.
Jeff, I have a question for you.
Yes.
You may be talking about this, but before we move on too far, is there any update on the ComD potential position?
Yeah. So, Jane, do you want to jump in here?
Sure. Yeah. So the
Interact.
ComD position. Yeah. So the ComD position is moving forward, but it's not quite as straightforward as a focused on the excellence initiative as the other ones. So right now, basically, there's a search going on for 3 different areas, one of which is autism. And so as you know, or maybe you don't know, for these hires to be approved and move forward, they require the commitment of the dean or deans of those respective, colleges to support the salary after the Excellence Initiative funds are done. And so the Dean of Public Health, there's a lot of complications around that. So right now, we don't have a clear path, that it's just an autism position being sought in the ComD department. It's just one thing they're considering. So that's kind of the way it's left at this point, is that it's just one of multiple. And if it works out that it aligns with the Excellence Initiative, great. And if it doesn't, then we can kind of revisit the option of having another hire that's more focused on the Excellence Initiative if the dean is open to that. So Jesse Kluzik can probably weigh in on that. And Jeff, I don't know if you have input as well.
Yeah, you summarized it as how I understood it, Jane. So
Okay, great.
We'll
Thank
see
you.
moving forward on it. We're hopeful. So, and Jane and I will keep pushing as much as we can. So the last thing I wanted to mention was in the first year, there were several of us that put together a P50 NIH grant for NIH on Fragile X syndrome, and that included Bokai Guoshi Kai, Fei Fei Zhao, Sophia Lizaraga, Fiona Hollis, David Mott, Abigail Hogan, Jesse Kluzik, Jane Roberts, and me. It wasn't funded, but it was an experience putting it together, and I think it was a good learning experience for all of us. And we're looking at ways to kind of revamp that and lick our wounds a bit on the reviews and where to go forward with that. So if you have ideas on that, let us know. But I think we put together a lot of the groundwork to be able to put together those large grants. So that will be good for our group moving forward. And then lastly was that we have an external advisory board now. And that is Gary Bissell at Emory University. He works on Fragile X syndrome and is a developmental neurobiologist. And in fact, he was a postdoctoral mentor for Christy Welshans. Marsha Malik at Wisconsin. I think it's Wisconsin or Minnesota?
Wisconsin.
Wisconsin, okay. Chris Cowan at MUSC, who I think most of you know. And then Joe Piven at UNC Chapel Hill. So, and we're going to meet with them in mid-November. Jane and I will. So So, as I mentioned, we'll have a meeting in December, and then we plan to continue these through the spring semester. Some of those are going to be the recruitment seminars, I believe, but other ones, if there are suggestions on how to do these meetings and what to do, that's the most useful for everyone, please let us know, because we want it to be, we want these to be of use, not wasting time. So, I have a good friend in Israel who refers to some things as chewing water, and we don't want to chew water. So, any
And I
questions?
just want to say thank you to Jeff and to Jane.
Oh,
You're welcome.
you're welcome.
Yeah, it's been very rewarding putting this together. So,
It has been. I guess I'll end on a somewhat positive note too, as many have sort of had made comments, but that is that even though we didn't get funded for that center initiative, there were a lot of really favorable comments and a lot of excitement about our approach to integrate across humans and animals and sort of integrate in this very challenging interdisciplinary way that we proposed. So to me, that's a lot of validation that the ideas behind this excellence initiative are recognized, you know, at the national level, and that we have a really good sort of start to trying to make this happen. And also recognition that it's so hard. You know, it would have been a miracle for us to get funded, but hey, miracles happen. And anyway, I thought we got some really great feedback. The other thing is that the provost spoke recently at a College of Arts and Sciences general faculty meeting and noted some of his priorities, and those include increasing research infrastructure, really promoting interdisciplinarity, and talking about sort of being a little more aggressive or assertive in terms of like fundraising and such. And I think that this excellence initiative really sits at many of the points of his sort of priorities. And you know, so I think that also bodes well just at the university level, that the kind of ideas that we all have and the work we're doing collectively is something that the administration seems very interested in independently. So Jeff and I will try to weasel our way in and make sure they see how supporting our work is supporting their work. And we'll keep you updated on that, and if you have ideas as well.
okay. Well, let us know if questions or issues or anything and suggestions. I guess we're actually done, and Jane's going to get to her next meeting, and so will Ruth. So,
Great.
thank you all.
Great. Thanks, everyone. It was great to see you.
Thanks, you
Good to
guys.
meet you, Layla.
You guys.
Thanks. Bye.
Bye.
Bye.